Because cancer is a cell growth abnormality, a good place to start the discussion would be on the normal cell cycle. Uh, you remember we have mentioned in previous discussions that the classical cell cycle goes from mitosis to the area between mitosis, which is called interphase. Interphase has three segments. The first segment is before DNA synthesis, so it's called pre-synthetic or G1. And then the third phase after DNA synthesis is pre-mitosis or G2. Anyway, the transitions between these four phases involve compounds called cyclins of various types, A, B, C, C1, D1, all these different types, as well as proteins called cyclin-dependent kinases, or CDKs of various types. And it's the combination of cyclins with the kinases that elevate and enable one phase of the cycle to transform into the next phase. This is the primary regulation on normal cell growth. Now, a discussion of cancer would not be appropriate unless you realize that there are inhibitory genes and processes and proteins to these cyclins and cyclin-dependent kinases. For example, there's an inhibitor called KIP or CYP if you're talking about a cyclin or a kinin inhibitory factor respectively. And then we have the INKs, and the INKs are the inhibitory of kinase, the inhibitors of kinases. Sometimes they're combined with another acronym named ARF. But just to remember, these are genes and proteins which are expressed, which inhibit these processes. So we already know that the inhibition of normal cell growth is a factor. In addition, we'll see that there is a whole variety of what we call tumor suppressor genes. And really, they are not tumor suppressor genes, they're normal growth suppressor genes, but somehow they got the name tumor suppressor genes. They don't suppress tumors, they suppress normal growth. And the massive and most widely studied gene in that area is P53. Let's now look at the whole process by which mutated genes become cancer-causing agents. And the best way to understand that is to remember that all of the genes in our body, or many of the genes in our body which do not make proteins, regulate growth or regulate DNA or regulate cell cycle in one way or another. And these are called proto-oncogenes. These are normal genes. Well, a mutation of a normal proto-oncogene is called an oncogene. And these are the abnormal genes now which make abnormal proteins called oncoproteins. And the five biggest categories of oncogenes would be genes which code for growth factors, genes which code for growth factor receptors, genes which are involved in signal transduction proteins. And a signal transduction is the process, it refers to any process in which a cell converts one kind of signal or stimulus into another. Or any process that regulates the nucleus is another category of oncogenes. And of course we saw the cell cycle regulators as well. Now if you hear the term oncoprotein, we're talking about a protein which is made by an oncogene. If you hear of an oncogene, you can think of it as a mutation of a normal gene. And the normal gene is called a proto-oncogene, and a proto-oncogene can generally be regarded as any gene which winds up coding for cell growth regulatory processes. This is the whole concept of oncogenes. So if you were to take the category of growth factors, for example, and we talked extensively about the growth factors, all the GFs, for example, the platelet-derived growth factor, fibroblast growth factors, transforming growth factors, hepatocyte growth factors. You know, these are all proteins coded by genes, which can be called cis, HST, TGF, HGF. Sometimes the proto-oncogene has the same symbol or three-letter acronym as the protein, and sometimes it refers to the oncogene. So just remember, when you see something like HGF, it could refer to the protein, the proto-oncogene, or the oncogene. And if these genes are either overexpressed or amplified, they may ultimately involve tumors in specific areas. For example, if you took TGF alpha, transforming growth factor alpha, and which is coded by the proto-oncogene TGFA, and it was overexpressed, well, it has been known that that process indeed does occur in some astrocytomas, or tumors of the glial cells of the brain. And that's the whole process for growth factors. Let's move on to the next category of growth factor receptors. Growth factors can't do anything unless they have receptors. So if you take these various growth factor receptors, whether you're talking about neurotropic factors, platelet-derived growth factor, EGF receptor family, well, their proto-oncogenes can be mutated, and as a result, they are either overexpressed or amplified or have a point mutation, a specific point mutation in the DNA sequence. And as a result, we know that these can result in specific tumors. So if you were to take colony stimulating factor 1 receptor, which is the proto-oncogene FMS, if that has a point mutation, it may result in a human leukemia. So these are examples of tumors like squamous cell carcinoma of the lung, breast and ovarian tumors, leukemias, which are the result of overexpression or amplification or point mutations of genes which ultimately code for growth factor receptors. And I want to tell you another thing. Whenever you see a chart that I present like this or this, and it looks like horrible, like, don't ever think you have to memorize it unless you want to. I think what you have to learn in some of these busy charts is not everything, but just the process. So in here, I gave an example of growth factor proto-oncogenes being activated and causing these tumors. And the next one, we talked about the growth factor receptors, and so on. Let's go to the next category of signal transduction proteins. Proteins involved in second messenger, perhaps. For example, GTP binding proteins, tyrosine kinase, RAS signal transduction. That's the big one, as well as another category called WNT signal transduction. These are all signal transduction proteins. They are coded by these genes, of which RAS, ultimately named because they were found in rat sarcomas a long time ago. And in most cases, a single point mutation of a gene which codes for a signal transduction protein can result in these tumors. And the P53 and the RAS oncogenes or proto-oncogenes, these are the single biggest and most widely studied oncogenes, and they are present in the majority of human neoplasms. But specifically, if the HRAS proto-oncogene gets mutated to be the HRAS oncogene, which is a GTP binding transduction protein, it may wind up as being a bladder or a kidney tumor. So in all of these three busy things which we just saw, the common denominator is that in every case we had a proto-oncogene which was mutated in some way to be an oncogene, winding up expressing an abnormal protein and ultimately winding up with the specific human tumor. And really, in hematologic tumors and many solid tumors, you can trace back processes like this. Okay, will we have time for the next one? No, we won't, but it's enough time to open the door. Let's just say we're going to talk about the class of nuclear regulatory proteins next, and I thank you very much.